So on behalf of myself, Bert Kosiski, also our president is joining us today, Jane Gunther, and our guest speaker, Fanny Elahi. We welcome you to the CADASIL Connection webinar series. cureCADASIL is a nonprofit organization dedicated to helping patients, families, caregivers, and other supporters touched by CADASIL. We aim to unite patients and the medical community toward the common goal of treatments and ultimately a cure for this rare genetic disease by promoting communication, advocacy, research, and education. We are indeed fortunate today to have Fanny Elahi presenting. She is a physician scientist in the Departments of Neurology, Neuroscience, and Pathology at the Icahn School of Medicine in Mount Sinai, New York. Her research interests include examining the link between disease of the brain's small blood vessels and neurodegeneration, and modeling disease using blood and cells from affected individuals to make discoveries with therapeutic potential and find a cure for CADASIL. Her talk today is the CADASIL Patient-Partnered Therapeutic Discovery Pipeline. The Elahi Lab at the Icahn School of Medicine in Mount Sinai uses a multidisciplinary approach to model disease, identify early biomarkers, and find therapeutic targets for vascular brain dysfunction and degeneration. They focus on genetic causes of vascular neurodegenerative disease to understand fundamental mechanisms that lead to disease pathologies in order to identify impactful therapies. They take a systems approach, combining blood and brain molecular analyses with clinical data to identify molecular pathologies driving clinical symptoms, and use patient stem cells to model mechanisms in a dish. In collaboration with cureCADASIL, the Elahi Lab and collaborators are assembling a US-based CADASIL cohort with banking of blood, plasma, white blood cells, clinical data, brain tissue, and patient-derived stem cells to build models of disease. The cellular models of disease will be used for screening therapeutic interventions.
Bert, you froze. Maybe try turning off your...
What froze? You're
good.
A major component of our collaborative work is
you might want to turn off your video camera.
Jane, I think it may be your connection because I can hear him.
Okay. I'll do that in any case. A major component of our collaborative work is to build a CADASIL Center of Excellence without walls, where work to advance mechanistic understanding of CADASIL happens in partnership with patients and their families. In this webinar, Dr. Elahi will summarize recent progress, highlight exciting future plans, and discuss different ways to get involved. So without further ado, I'm going to hand it over to Dr. Elahi.
Thanks so much, Bert and Jane. Thanks, Bert, for that introduction. Can you see my screen? Let me just make it big. Can you see my screen?
Yes, it's perfect.
Okay, wonderful. So it's a real pleasure to speak with you today. I wish that this was in person. I wish that this was interactive, but we'll make the best of it. And what I've done is that I've organized my talk in modules. Not gonna have time to show all the data. I do wanna give you a flavor of the spectrum of the work that we've undertaken to find therapies for CADASIL. And then I want more time for discussion and questions. And I hope that this will be one of the many interactions that we'll have on this important topic. So I want to start with a thank you slide because I don't want to run out of this out of time and not be able to thank everyone. So first and foremost, I really want to extend deep and heartfelt thanks to the research participants and their partners and families who have trusted us with their time and come and participated in the research that we're doing, donating blood, allowing us to image their brains, doing the gruesome cognitive testings that we ought to do. I also want to thank my incredible lab. It's really fun and rewarding to work with incredible minds who are so dedicated to therapeutic discovery in CADASIL. And then our collaborators, it really does take a village. That village is composed of patients, participants, lab members, and incredible collaborators from across the US. I've just named a few, there are more. If I had to put all the names, I think I could fill an entire slide. And then our funders for funding the work to move this knowledge forward. So this is what my lab does. We don't have animal models. We think that the initial discovery really ought to happen in humans. And now we think that we are equipped with the tools to identify important molecules and cells that are contributing to the clinical abnormalities in CADASIL, and use this as a launch pad really to then look at mechanisms in various kinds of models. We do stem cell derived human models. There are also good animal models out there that other labs and our collaborators are using. So we do this not just for CADASIL, for all neurodegenerative diseases and vascular disorders, because we're interested in finding commonalities which would allow drug repurposing, and also would connect researchers working on CADASIL to research out there on other disorders. So we have brain tissue, we have plasma, we have brain imaging, we have clinical phenotyping, and then through the generous donation of research participants, we collect cells, we turn them into stem cells, and then we differentiate them into various kinds of primary cell types to model disease in a dish. So this is the outline. I'll talk about CADASIL, some things that we know and some things that remain a challenge. And then about the concept of endophenotype. This term refers to clinical or I'm using it in a broad sense, imaging or molecular footprints that are associated with genetic variation, in this case, mutations in the gene Notch3. And we think that these endophenotypes are really critical to allowing us to study disease early. And I think, as we all know in the field, whether we're studying CADASIL or not, early detection and understanding the earliest stages of disease are really critical to therapeutic development. And then I'll make sure that we have time for the discovery pipeline that we've put in place that I would love to discuss with you, and would love to have anyone who's interested in participating in that. So this is an overarching view of what we hypothesize is happening to the brain when vasculature are dysfunctional. It doesn't just pertain to CADASIL, but we think CADASIL may be a bigger effect size for the vascular abnormalities. Despite the fact that we've all been very focused on the brain, there are abnormalities systemically in blood and other tissues that may actually begin before brain abnormalities are detected from a clinical perspective. So there's a component of inflammation, changes in coagulation. Then there's this vascular barrier protecting the brain from anything that happens in one's body. We think that becomes dysfunctional. Downstream of that are all sorts of changes, either to the matrix of the brain, the tissue, basically the molecules surrounding the cells, and eventually the cells themselves in the brain. And that leads to dysfunction and degeneration. And that's really where clinical symptoms begin. So to sort of walk backwards, this is showing disease of small blood vessels as a starting point. There are all sorts of molecular abnormalities ensue. There are imaging abnormalities that can be detected more sensitive. And then the less sensitive, but sort of classical abnormalities are the white matter changes, which anyone who's affected with CADASIL has heard at some point when they did a brain MRI. What we want to do is walk backwards, start with the imaging abnormalities and work out the molecular and cellular underpinnings of these imaging abnormalities. What's the challenge? Well, the challenge is that disease is very heterogeneous. When we study humans, we're not actually capturing the full movie as disease develops. It's really snapshots in time. And so we need to be very clever about the design of studies in order to be able to uncover longitudinal pathology, sort of the story of how abnormalities start, how they evolve, and when is it that we must intervene. I'm going to give the example of stroke. Stroke has the great advantage. Jane, I think I hear some background noise. It may be coming from your mic. Do you mind muting? I don't know. I hear some background. Thank you. So stroke has an incidence, which means that when someone has a stroke, starting at that point, they can be studied just so that we understand how pathology evolves and what follows what, and sort of get this longitudinal story. Obviously, some individuals with CADASIL have strokes, but not everyone. And it's unclear whether stroke occurs early or not. We don't think it's the earliest manifestation of the disease. And so we don't want to start studying disease after a stroke. So what do? This is sort of what we're facing. Basically, CADASIL has a lot of changes that have been described, including on pathology. We know a lot of different molecular and cellular changes. And what I mean by pathology is when someone dies and we look at the tissue. With imaging and using animal models, we know that there's a decoupling of vascular function with neuronal function. So as we use our brain, blood flow changes to demand of the brain, and there's a dysfunction in that signaling in individuals with CADASIL. Then we know that the covering of the neurons, the myelin changes and degenerates, and ultimately there can be dysfunction of cognition. So what we call cognitive impairment. Some things that the individual was able to do can no longer do. But what we don't know is what follows what in the human brain, and when can we intervene in order to stop this cascade. When we look at a human, they've had decades of this occurring. And so what we want to do is to start studying the earliest stages of disease, but also continue to study all the stages of disease and sort of create this, what I call a pseudo longitudinal, as though we do have a film of the pathology developing across multiple individuals at various stages of disease. This is published by colleagues in France. As you may know, that's where the gene was actually discovered for CADASIL, and they've studied hundreds of individuals in their cohort to sort of sequence the abnormalities that can be detected. While this is extremely helpful, I think this may not be the case for everyone. For instance, in the individuals that we have studied in our cohort, not everyone has strokes, and the mood disturbances can actually happen much, much earlier. In some ways, sequencing clinical abnormalities really depends on the tools that we have and how sensitive they are for detection of these abnormalities. This is from the same group, and they've described beautifully the pathology on brain tissue. Others in the US and elsewhere have also done that. So we know some things that happen to the blood vessels. We also are getting hints as to what we can look at on brain imaging. And as I mentioned earlier, the white matter changes on a specific sequence are really classical for CADASIL. I'll bring your attention to these spots, these spaces here that we call perivascular spaces, because we have some data that I will share about this in a few slides. So this is the gene, and these are, and here I'm showing you the quote unquote hot spots of mutation. For reasons that we don't fully understand yet, there is a clustering of mutations in the first section of the gene. And it may be in some ways that these regions give rise to more pronounced symptoms, and so they're more frequently detected, and that there are lots of mutations that go undetected, and there are a lot of people living with CADASIL with perhaps very mild symptoms that don't know that they have a mutation in Notch3. And in fact, now we know that that's the case. If you look at the clinical incidence of disease, so this is people with symptoms that have been diagnosed with CADASIL and Notch3 mutation, it's rare. So it is a rare clinical disease. But we know through a study in the UK Biobank that the frequency of the mutation is much higher in the population. It's just that people are not getting diagnosed with a clinical disease. And I think that brings up the question of what are the first symptoms? And I'm going to broaden this to say, what are the first abnormalities that we can detect? And can we screen for that at the population level? So it can't be really complicated. It needs to be something that we can screen for. My lab is really focused on molecular markers. So we hope to have a molecular marker for which we can screen at population level. And this is really where we stand. We have knowledge of some molecular abnormalities through various tools and model systems. We have knowledge of pathological abnormalities when someone dies. What we lack is how do we go from a mutation all the way to the plethora of pathological abnormalities and symptoms and the constellation of symptoms, syndromes. This is what we hope to unravel with the CADASIL patient partner discovery pipeline. So we have a clinical cohort. This clinical cohort started at UCSF in collaboration with clinician researchers such as Michael Geschwin, Joel Kramer, and others. And we started studying individuals across the spectrum of age and symptomatology. All that we wanted to know is that someone had either a genetic mutation, a family history, or imaging abnormalities that were suspicious for CADASIL. As young as 18, as old as however old an individual is before they pass away, and then the full spectrum of function and cognition. This study has now moved to Mount Sinai, and I've put the contact information if you are interested in contacting us and enrolling in the study. We also have collaborators from across the US, and I'll highlight Dr. James Meskia at Mayo Clinic. So if that's a closer site to you, we can connect you to his program. And I also want to thank Dr. Luana Murray and Ryan Park, the clinical research coordinators who are leading the enrollment and data acquisition for this study. So here I'm going to show you the spectrum of demographics, so to speak. So this is the spectrum of age. You can see really we do cover the full spectrum of age, lower numbers at the tail ends. This is the systolic blood pressure. So many of individuals in our research cohort don't necessarily have hypertension or other vascular risk factors. They just carry a mutation. And then cognition is also by CDR box score. It's also quite wide, but as you can see, a lot of individuals early in their symptomatology. This is the white matter spread, and for sake of time, I'll go forward. So what I want to show you is how diverse the spectrum of disease can be. Here is images from someone in the cohort who has vascular risk factors, is a heavy smoker, and has had a hard life. This individual has had multiple TIAs, and as you can see, there's a lot of white matter changes on the scan that can be seen. But I'll also bring your attention to the fact that there really isn't a lot of volume loss on the brain imaging. And that means a lot, because we think that once the atrophy of the brain starts, that's when a lot of the symptoms arise and progress. This is another example. So here you do see volume loss. And this is another one. You see some volume loss, you see white matter changes. And then you can start seeing these white matter changes as early as someone's 20s, maybe even teens. We haven't scanned anyone before age 18, but you can see that the brain otherwise looks really fantastic. So it's just these white matter changes. And this individual had really no symptoms. Similar. And as we started studying individuals in this cohort and scanning their brains and collecting the data, we were struck by what I call outliers, but perhaps these individuals are not that far and few in between. And on a larger scale, if we scanned more people and studied more people, we would find more individuals who seem to be protected from the effects of Notch3 mutation. And so here, there's a striking image of an individual whose brother had severe disease in his 40s, and this individual had 0 symptoms and strikingly no imaging abnormalities well into his 50s. So same mutation, same family, completely different outcomes. There are some things that we are looking into that are giving us hints of protective factors, but we really want to study this at a larger scale, because this is as much an opportunity for therapeutic discovery as understanding what the pathology is about. So protective factors are incredibly important. Same here, an individual in her 40s and really no imaging abnormalities. So these are sort of what we are using as the classical markers of disease. There's white matter changes, there's spaces, these spaces that I showed you on the brain tissue, the perivascular spaces, and you can see here as white dots in the middle of the brain scan. These are the classical markers that we're studying. I would call them structural markers, and there are a lot of new markers on imaging that arise, that are coming about. But here's what I want to highlight. So you have these imaging abnormalities at macro scale, and then what we're thinking in our research is, what are the cellular underpinnings of these imaging abnormalities? That's what we need to know in order to be able to unravel the biology of disease. So I'll show you some, and so here is a reminder of the pathology abnormality seen these perivascular spaces. And on brain imaging, they show up as these sort of spaces, elongated spaces. These are spaces around blood vessels, typically veins, but arteries can also have enlarged spaces around them. And we think this is the route for drainage of various kinds of things that the brain wants to get rid of. So there's a way for us to quantify these on imaging, and here those colored spots on the right scan are these spaces that we can quantify. And what's interesting is that if you quantify these spaces across diseases, there's a lot of overlap. So normal individuals can have a lot of enlarged perivascular spaces, and individuals with disease could have, to a lesser extent, enlarged perivascular spaces. So this kind of imaging abnormality is, I'll take that back, I can't even call it abnormality. This kind of imaging and phenotype is not as simple as the white matter changes that I showed you earlier on. There's a lot more that we need to understand about these perivascular spaces. This study led by a student in the lab, and now he's a research assistant, Bradley O, and a colleague, Tori Sui, and the imaging team at UCSF was done over the past 2-3 years. And now we're in the advanced stages of working on a publication. We're not the only ones who've studied the perivascular spaces in CADASIL. There have been 2 other publications already, but we are studying this, I think, in the earliest stages of disease. And what was really interesting is that in the green line, I'm showing you normal individuals, no Notch3 mutation, and in the pink reddish line are individuals with Notch3 mutation across the spectrum of disease symptomatology. And here you see that on the x-axis and on the y-axis, basically on the x-axis is the perivascular spaces, on the y-axis are the white matter changes, the classical finding, and the association is really strong in CADASIL, but not normal controls. And so the perivascular spaces mean something in CADASIL specifically. What it means, we don't know, but because of this result, we're going to continue to study the perivascular spaces. And as I'll show you in future slides, we have a way to run correlations with molecular markers to understand what's driving these imaging findings. Another thing that we did is study the retina. It has the distinct advantage of being non-invasive. You can do it easily. It takes minutes to do that for anyone. So if there's anything that we can detect on retinal scanning, then you can imagine it can help in risk stratification for clinical trials. And ultimately, when we have therapies that we're going to take to clinical trials, perhaps serve as a biomarker of therapeutic efficacy in clinical trials. A lot needs to be done before we get there, but we wanted to explore whether there's any significant differences in retina of individuals with CADASIL and controls. And so this was led by a postdoc, Dr. Ahmed Abdelhak, in a collaborator's lab, Ari Green, who does retinal imaging in multiple sclerosis patients. And here I'm showing you basically a large vessel in the retina. That red line is showing a large vessel, and there's a way to measure the vessel walls of various kinds of vessels. It's targeted, so you choose one vessel, you could choose 2 or 3 vessels and take an average, but basically these are the larger retinal vessels. These are not the capillary or the smallest of the blood vessels in the retina. And so we measured the vessel wall thickness, and what we found is that here on the graph on the right, I'm showing you CADASIL, vascular cognitive impairment without a Notch3 mutation, and then various kinds of groups. And you can see that these individuals who have CADASIL are people in our cohort. So they're early symptomatic disease or no symptoms. But despite being really early in their disease, we do see that their vessel walls are thickened. This was, I would say, a good surprise because, again, if we can scan retina, it is really going to be helpful. It's cheap, it's fast, it's comfortable for the patients. And then what we did on the left, in collaboration with researchers at Rush University, we looked at a measure on brain imaging, arteriolosclerosis. This is a measure of abnormality of small blood vessels of the brain, and showed that the vessel wall thickness is correlated with this measure of arteriolosclerosis on brain imaging. This, all these findings are published in Alzheimer's and Dementia Journal. Other brain imaging modalities that we're investigating in the future, there's this vessel wall imaging that we think can capture vascular inflammation. And so you give contrast and then you measure various kinds of blood vessels in the brain and see whether there's this phenomenon called enhancement, where the contrast is retained in the vessel walls as a measure of, we think, endothelial and vascular inflammation. There's also transcranial Doppler and EEG measures that we can capture. And so this is looking at the electrical activity of the brain in tandem with blood flow. And we think because of this neurovascular decoupling that happens in CADASIL, this may be a promising imaging modality to investigate. Another imaging modality that we are investigating in the future is the leakage at the blood-brain barrier. So if you remember the first set of slides that I showed, we think that this communication between the blood and brain is disrupted in diseases of small blood vessels, and we want to know whether this is the case for CADASIL. There are two publications in mouse models. One said yes, and one showed no. And so we want to know what is happening in humans and if there is leakage, at what stage of disease there's leakage. This kind of phenotype has a very clear set of therapeutics that could be applied if there's leakage. Another imaging modality is diffusion measure. For sake of time, I'm going to go forward, but this also we're investigating. And then now onto the heart of what we do in the lab. We use everything that I showed you as what we call a clinical data point, whether it's clinical symptoms, cognition, or imaging. We think these are phenotypes. And what we are really interested in doing is understanding what are the molecular and cellular underpinnings of these phenotypes in humans across the spectrum of disease severity. And so here, we have imaging, and here you can replace this by cognitive testing, basically anything that is collected from living humans. And then we do a bunch of things in the lab, including looking at molecules in blood, in cerebrospinal fluid. We don't have a lot of cerebrospinal fluid, but we could do it if we had it. In things called extracellular vesicles. These are vesicles that are secreted from various kinds of cell types into blood or cerebrospinal fluid. And then also collect cells, make the stem cells that I mentioned, and then study abnormalities in a dish. And then we are looking at any kind of molecule that can give us a sense of the molecular pathologies, protein, RNA, lipids, metabolites, etc. So these are some of the faces behind the work that we've undertaken. Dr. Stephen Fitzsimons is an incredibly talented postdoctoral scholar in the lab. Jonah Keller is a very talented undergraduate student, and Dr. Lisa McDonald is also a talented postdoc in the lab. So Dr. Stephen Fitzsimons and Jonah Keller are analyzing what we refer to here on out as unbiased proteomics. And so this is basically, we're using a technology that allows us to measure 7,500 proteins in any kind of biofluid, and we're doing plasma in this case, and then run analytical pipelines in order to uncover molecular abnormalities, and then link that back to cells of origin. And then what we want to do is, once we have those sets of abnormalities, build panels of specific proteins that we think are important to the various kinds of pathologies that we want to characterize at large scale, and then put that on an automated platform, such as the Simoa Quanterix that we have, and study it at scale. Another approach is isolation of cell-specific extracellular vesicles and looking at the content of these vesicles. And we're doing all of these in parallel. I'll skip this. I've already talked about the overview. So on to some results. And I'm not gonna show the full extent of results that we have generated for sake of time. But this work is really underway in late stages of preparation, and so hopefully we'll have the full set of analysis done soon, and would be delighted to share that in form of publication and perhaps another webinar. And so here, when we look at the 7,500 proteins, we look to see what's high in levels or low in levels in comparison to individuals who don't carry a Notch3 mutation. That's the very first step of our analysis. And I must mention that this work is also has been made possible through a collaboration with Dr. James Meskia, who's given us samples from his cohort. And so here we look at what's up and what's down. And then this is showing you a different way to visualize. And what's really striking here, I would say, is how we are seeing clear footprints of CADASIL versus controls. And so on the left, you see CADASIL, on the right, you see controls, and you can see that there's a lot more similar coloring on in each group. But I also want to bring your attention to outliers. So, you know, where you see red for CADASIL, you also see some blue, which is similar footprint to controls. And so we are really interested in understanding both what the abnormalities are, as well as what are the factors that are protecting an individual from developing the signs and symptoms and molecular abnormalities associated with their mutation. So for sake of time here, I'm going to skip a bunch of things because I want to show you the all-in-human discovery pipeline. Here, this is where I want to end. So I've shown you a bunch of tools that we want to combine in taking this work all the way to identification of specific drugs, interventions, molecules that we can give to individuals affected with CADASIL, either symptomatic or just carrying the mutation, to prevent disease progression. And ultimately, the dream is to cure disease. So having individuals with a Notch3 mutation never develop clinical symptoms. And how we want to do that is through a partnership with patients and their families and partners and advocates to bring individuals into the research and various kinds of locations for this, collect blood, get some imaging. Imaging is, I would say, not the most critical, but blood really is. And then drive the stem cells, build various kinds of models of stem cells to, in a dish, study the molecular and cellular pathologies, use the human tissue from post-mortem brain tissue, as well as the blood and cerebrospinal fluid molecular studies, where I showed you some examples. Make sure that what we have in a dish is reflecting the pathology in living humans, and then use the tools that are available to any disease, cancer, other neurodegenerative disease, immunological disorders, to basically screen whole libraries of molecules and interventions against these dish models in order to identify therapies that either stop, reverse, or reestablish the normal physiology for CADASIL. And so this really does truly take a multidisciplinary team, and I'm extremely excited that cureCADASIL has helped us assemble such a team in order to tackle a project at this scale. Again, if you're interested in participating as a patient, as a family member, or as a researcher, as a clinician, please reach out at studies at vaskbrain.com, studies@vaskbrain.com, or just send me an email and I'll connect you to our study group. Ultimately, this is what we want. I'm just putting some things on there, but really we don't know what starts, what follows. We want to get the full set of pathologies on a time scale of this is the first abnormality, this is how we detect it. This is the intervention to have if someone is presenting at this stage of disease and at a later stage and maybe a different intervention, we hope that this will be the deliverable from the kind of project that we have designed. And in conclusion, CADASIL is heterogeneous. What I mean by that is that it doesn't present as one disease. It's a spectrum of symptoms and clinical severity. And the molecular underpinnings of that are also going to be diverse. We want to find what gives us specific diagnostics for the various kinds of subtypes, in addition to finding shared common therapeutic entry points. We're also extremely interested in understanding the protective factors, because we can leverage that for the discovery of therapies that could apply to potentially a wide range of symptomatology. For therapies, we need modeling of disease in humans living with this condition, as well as deriving dish models from those humans living with this condition. And ultimately, as a physician scientist, I am convinced that our discoveries are only going to be possible in close partnership with patients, their partners, and all stakeholders. We think we understand disease, but every time that I meet a patient, I am reminded that they're really the expert for the clinical symptomatology of this disease, and we need to understand what are the first changes, what are the first abnormalities, also what bothers patients most, because we could work on something that ends up being tangential to what it is that they really want to be treated first. I mean, it's fantastic if we have one therapy for all symptoms in CADASIL and ultimately cure it, but before we get there, it's possible that we're going to have various kinds of therapies that can help different aspects of the disease. So if you're compelled by this study, if you're interested in participating, please don't hesitate to reach out at studies@fastbrain.com or send me or my research coordinators an email directly. I'll stop here to allow time for questions and discussion. Thank you.
This has been great, Fanny. Thanks very much for your presentation. And there are some questions, and I have some questions I can ask too. But first, I'll remind everyone that you can type questions in the question box. And so if you think of other things as we go along here in the discussion, please ask your questions. So a couple of questions that have come through. One is somebody is asking, how many participants are there right now in your study?
We had to pause for a bit when we moved from UCSF to Sinai, and I believe we have 36 participants who have come in and participated in person. And I should add that in order to allow more individuals to participate in our research, we have also built what we call a remote cohort. So that means that you can participate in the research by remotely from your home and never come in to see us. We send a mobile phlebotomist to your home to collect the blood, and then we do the cognitive testing over the phone and through an app on a smartphone. And so I think participants in the remote cohort are closer to 40-something, 50. And every day we have more individuals joining, so.
Another question was, here, my daughter was diagnosed at age 10 with some non-stroke-like symptoms and prompted in imaging and genetic studies. Would she be able to participate with parental approval in your study?
Can you repeat? Daughter is how old?
Yeah, age 10. So a minor who's not legal age yet, can a minor participant with parental consent in your study?
That's a very, very important question. I would really like to make that possible, and we will work on making it possible. As it stands, our regulatory approval, our ethics board approval is only for individuals 18 and above. but there has been precedent. There is another study of genetic neurodegenerative diseases that is now enrolling children, and we want, with their help, do the same for CADASIL. So as it stands, we're not able to study minors, but as soon as we get approval from the ethics board, we will let you know. So do send me an email, and then we will let you know as soon as we're able to study your daughter.
Here's another more technical question. Were the proteomics data from patients with symptoms? Can you stratify the proteomic signatures by the symptomatic state?
So we don't have enough individuals across the full spectrum. This is really a start, and the results are so fantastic that we want to really scale this up. So we're just limited by how many people we've collected samples from. We do think that there may be a change in signature across the disease severity, and this is something that Stephen Fitzsimons is actually currently looking at, but I think we're not going to have the full power we need. So we need more blood samples from individuals super early, maybe even pre-symptom, and then as they develop symptoms. But for Alzheimer's disease, we did this, and there definitely are signatures of disease severity. So in other diseases, there has been precedence. We just haven't yet looked at this in our CADASIL cohort.
So here's a good follow-up question to that. How many research participants do you want to have to be able to effectively do your work? What's your sample size? Goals.
I'll tell you that, yeah. So I'll say that we've already been able to make some discoveries based on our sample size. But I want hundreds, because I think that we need to be really sure, and we need to look at more. So we need, I think, hundreds. I would say 200 would be fantastic. That give us enough individuals at the various stages to be able to look at all the different aspects of the molecular and cellular abnormalities that we want to. And because CADASIL is so diverse and so heterogeneous, I think, 200 or so, and the more the merrier, I would say, would be ideal.
Someone asked, and I think this is an interesting question, given all the variability that it's out there clinically, are we missing an interaction somewhere? Is there something there that we're missing in all the analyses that have been done? People have looked, of course, at cardiovascular risk factors, smoking and cholesterol and whatnot, and there is some, higher risk with that, but it's really nothing to write home about. So are we missing something out there? And the questioner here asks about diet, exercise, supplements, any of these things out there that might be protective or vice versa.
So we already know that the vascular risk factors, and I'm going to mention hypertension as the predominant vascular risk factor that interacts with mutation status. And we know this on larger scale through clinical studies, but also I'll say in my clinical practice, I see that individuals who have had a very healthy lifestyle have discovered that they have a mutation in their 60s, completely asymptomatic before that. So even anecdotally, I see that it's the case. And I say that, and I think the anecdotes are actually important because it speaks to the effect size, right? So you have these studies and you can have hundreds of individuals in the study, but when you see that manifest in your clinical practice at an N of 1 level, so one individual, you see that there is, that the effect size may be large. And the same is true for, I would say, how I think it's protecting people. It's in the same families. Lifestyle really does diversify symptom severity. smoking is bad, some of the recreational drugs can be awful, cocaine and the families of that. And then exercise in general, we think is really important for vascular health. And in this case, I'm intrigued by the enlarged perivascular spaces, and there may be some clearance abnormalities. And we think, again, that exercise is really important for brain clearance. Now, but all of these things need to be shown more clearly, I think, and more convincingly. What's really intriguing to me, and this is not just for CADASIL, it's for any kind of brain degenerative or brain disease, is diet. I think we say Mediterranean diet, but as both a physician and a scientist, I'm unsatisfied with that answer to a certain extent, because I think that diet is going to be more precise than that. I think we just need better tools. And again, I go back to these molecular biomarkers in order to be able to determine more precisely what combination of diet is fit for one person than another. And here I think of the microbiome research that we are not doing, but others have undertaken in Alzheimer's disease, Parkinson's disease, and other disorders. There's a very strong connection between the gut and the brain. And I think diet impacts the gut microbiome. So more to be uncovered there. And perhaps this is why I use the word therapeutic and not just drug, because I think a lot of the lifestyle interventions could also be very helpful. And it may, treatment ultimately may come from a combination of lifestyle and molecular therapies. So this is my long-winded answer.
That's great. There are a lot of questions rolling in here, and we'll keep going.
Yeah, yes. I'd like to bring up a couple of questions that came in that are about your study, because I think there's one in particular I'd like to have some clarity on. And the question is this the same study that is happening at Columbia with Jose Gutierrez and Fanny? I think you can be able to address that question, but if not, I can add some information to that.
Yeah, it's not the same study. There are a lot of sites enrolling CADASIL patients and collecting data, and I am also collaborating on that study with the co-PIs, Jane Paulson and Michael Geschwin. I think the studies can be very synergistic, and we are collecting samples, we're just not collecting cells. So we still want to have individuals come into either our site or James Mesquia's site in order to donate the cells. That's what's, I think, missing from that study. It's possible that in the future we'll be able to implement that, but as of yet, cells are not being collected separately.
Yeah, and I'll just add for people to understand the two studies, there's the CADASIL Consortium study, and then there's Fanny's Fast Brain, and you can participate in both studies.
Absolutely.
In fact, we want you to participate in both studies, because getting this data, the sooner we can get this data, that's really just driving towards getting to therapeutics. So definitely patient participation is greatly needed. So other questions that came in about participating is, are you interested in enrolling family members even if they don't have symptoms yet? Absolutely. Or at all?
Absolutely. In fact, you don't need to know. So our study doesn't require you to know your genetic status. And in fact, if you never want to find out whether you have a mutation or not, but are still interested in participating and helping this research move forward, you can. So family members at any stage are absolutely welcome. And I think it's very important to capture the full spectrum. Yeah. And then if you end up not having a mutation, then you just serve as a control, because we need individuals from the same families who don't carry the mutation. That's really the ideal control group.
Great. And another question about participating is, can one participate anonymously? And their concern is in terms of health insurance.
This is fully anonymous. So, and I can have a longer conversation. Feel free to reach out and we can have a longer conversation. I can explain exactly what we can do to to ensure that you don't get discriminated against.
And this question comes from a physician or clinical researcher. In my institute, we treat patients with neurodevelopmental or disorders. Would they be eligible for the research?
Are these individual 18 and above or minors?
Yeah, and maybe that's a good conversation for you to have.
So please reach out and we can have the conversation. I think going back to the initial question, we're extremely interested in extending the start of the study to minors, but there's a lot to be considered there. And so we want to do that very carefully.
Can you say against somebody is asking what email address can they contact you with about enrolling in the study? I think you gave that, but can you?
Yeah, I can go back to the slide. So both my email address, my coordinators, and then this. Are you able to see?
There we go. Yeah. And while people are writing down that address, a participant said they said that I'm a participant. I want to strongly encourage people to participate in the study. It's a very well organized and productive study.
Thank you so much. And we're so delighted. And I wanted to again thank the research participants and their families, and also cureCADASIL for making this possible, for connecting the community and making such research possible.
And we're very thankful to you and your lab for the terrific work that you're doing. It's very leading edge. It's really driving towards understanding the disease, which then will, as you had well laid out, will drive towards finding disease-modifying therapeutics.
Thank you so much. And if anyone has more questions or wants to discuss anything, please reach out. Don't hesitate.
No, it's top of the hour, but just a couple of other quick questions I'm scanning down that I think are very interesting. And one is, if you don't mind, one is, do perivascular spaces appear in other disorders?
Yes.
And are direct cause risk factors known for these perivascular spaces?
We are conducting a study, a multi-site study with researchers focused on Alzheimer's disease and other neurodegenerative disorders. And absolutely, perivascular spaces are a feature of the brain. And in fact, you can see them developmentally as well. So we're really beginning at the beginning stages of understanding what they represent. But I think from studying all these different disorders in our multidisciplinary team, we're realizing that they mean different things in different diseases. So perivascular spaces mean something in CADASIL that is very different from Alzheimer's disease, potentially. The other aspect of perivascular spaces that are important is their dynamic. So if you scan someone in the morning and at night, they can have different sizes of their perivascular spaces. So this dynamic feature of perivascular spaces makes it a very interesting and intriguing imaging marker.
One final question, because I know we have to get off, but this is a good question. Be careful how you answer it. Are you seeing patients at Mount Sinai?
I am, and I would say it's my joy. I think doing the science is fantastic, but seeing the patients is the reward. So I do need to dedicate a lot of time to research, but I've reserved my my clinic for CADASIL patients specifically. You are VIP, and then anyone with white matter changes that of unknown etiology, and some of them end up having CADASIL, and some end up having other causes for their white matter. So if you contact us, we can also connect you with my clinical admin to schedule a visiting clinic.
Excellent. So I think we'll end. I'm going to take back the screen for a final slide. So yeah, I'd like to thank everyone. This has been great. Especially I want to thank Fanny. It's been a great presentation and raises a lot of very intriguing prospects for the future. And so I'll remind everyone that there will be a recording this webinar that will be available online. And we hope to have more webinars very soon. There's a lot of activities going on within our organization, and I think there'll be a lot of spin-off webinars from that. And you can, of course, visit us online at the address that you see on this slide. And so with that, I think we'll end this webinar. And again, thank you very much. And thank you, Fanny, and all of the great participants and great questions that came out of this. Thank you.
Thanks so much.